In this video, we are going to go over different types of PCR. I recommend you start with a standard PCR first, but if you already know how it works, feel free to skip to the type of PCR you're interested in. As I said, let's start with a standard PCR. PCR, or polymerase chain reaction, is a very powerful laboratory technique that is used for copying or amplifying parts of DNA. To amplify DNA, we need a few things. An enzyme that does all the copying, and that is, of course, DNA polymerase, which you should already know from DNA replication, for example. DNA polymerase needs primers to work, and because we want to copy DNA which is double-stranded, we need one primer that is complementary to the 5' end of one strand, and another primer that is complementary to the 3' end of the other strand. We also need 4-deoxyribonucleoside triphosphates, DNTPs, which the DNA polymerase will use as building blocks for the newly synthesized strands. Finally, we need a buffer which creates a suitable environment for the reaction and of course the DNA from which we want to copy. Now that we have everything ready, let's take a look at the process in detail. A typical PCR consists of 3 reactions, or we can say a chain of 3 reactions that are repeated in multiple cycles. For that reason, the technique is called polymerase chain reaction. The first step in the reaction is to separate the 2 strands using a process called denaturation. We simply heat the DNA to about 90 degrees Celsius, which causes the 2 DNA strands to separate. Once the 2 strands are separated, we can proceed to the second step, which is called annealing. During this step, the temperature is lowered to about 40 to 65 degrees Celsius, and the primers attach, anneal, to the target DNA sequence we want to amplify, we want to copy. In the final step, called extension, we again heat the mixture to about 70 degrees Celsius, which allows DNA polymerase to synthesize the new DNA strands in the 5' to 3' direction. At the end of the cycle, we get 2 double-stranded DNA sequences we wanted to copy. Each cycle only takes about a minute, so you can imagine how much DNA can be copied in an hour. Before we move on, we should also mention 2 important things. First, because the temperatures in PCR change rapidly, we need a special kind of DNA polymerase that is stable at high temperatures. For that reason, we use special polymerase called Taq polymerase, which was originally isolated from a bacterium that lives in hot springs. The second thing is that we use special machines for the reaction, which are called thermal cyclers, and which are capable of changing the temperatures that we need for the reactions very quickly. So this is how a standard PCR works, and now let's take a look at the first modification. Quantitative PCR, or real-time PCR, has one important advantage. It allows us to monitor the amount of the amplified product in real time. To do this, we use a special kind of thermocycler that has a laser that illuminates PCR reaction tubes, the tubes in which the reactions are taking place. The DNA in the tube then emits a fluorescent light, which is captured by the detector, and the information is sent into a computer. The more fluorescence measured, the more DNA in the sample. That is the basic notion behind this method. But how do we make the DNA sample emit a fluorescent light? There are 2 commonly used methods. The first one uses a special dye that binds to the DNA, such as cyber green. After extension, the DNA binding dye then binds to the newly synthesized double-stranded DNA product, which as a result now emits fluorescent light. The more product there is, the more fluorescent light is emitted. The second method uses a dye-containing probe, for example a Taqman probe. A probe is a short sequence of DNA that contains 2 important parts, a reporter that emits a fluorescent light, and a quencher which blocks the reporter. This is important because we want the DNA to be able to emit fluorescent light after we have amplified the product, not before. In annealing, both the primers and the probes anneal to their complementary sequences, and in extension, as DNA polymerase synthesizes the new DNA strand, it cleaves the probe, which separates the quencher from the reporter, and as a result, the reporter starts emitting fluorescent light. And once again, the more DNA product we have, the more fluorescent light there is. The third type of PCR is RT-PCR, reverse transcriptase polymerase chain reaction, which allows us to use RNA as a template by using an enzyme called reverse transcriptase to transcribe RNA into cDNA, the so-called complementary DNA, which is then amplified using the standard PCR method. Reverse transcriptase is mainly found in retroviruses, for example in HIV. The components of RT-PCR are the same as in standard PCR, apart from the reverse transcriptase and an RNA template. To carry out an RT-PCR, first a primer must bind to the RNA molecule, which allows reverse transcriptase to synthesize a new cDNA strand. The original RNA is then digested by another enzyme, RNase H, and a second cDNA chain is synthesized by DNA polymerase. Then the reaction continues as conventional PCR. Today, perhaps the most well-known application of RT-PCR is the diagnostics of diseases caused by RNA viruses. To do this, the viral RNA is transcribed into cDNA by reverse transcriptase and amplified after complementary primers bind to their target sequences. The amplified viral cDNA then confirms the viral infection. Another modification is nested PCR. What can sometimes happen in standard PCR is that the primers we picked were not specific enough, and that some of them bound to a different sequence we did not want them to. This creates what we call a non-specific product, a PCR product that does not contain the specific DNA sequence we wanted. So the whole point of nested PCR is to get rid of these non-specificities, and the key to doing that is to use two sets of primers. Let's take a look and see how this works in more detail. Nested PCR consists of two PCR reactions. Each reaction uses a different set of primers. In the first PCR reaction in annealing, we use the so-called external primers. External primers are primers that are designed to be complementary to a sequence that is outside the DNA sequence we want to amplify. Of course, it is still very close to it, and it includes the sequence of our interest. By doing so, we get a slightly larger product in extension when DNA polymerase synthesizes the two DNA strands. Why do we do this? Well, imagine now that the external primers we used were not specific enough, and that we now have a mixture of the DNA sequence we wanted and of some other DNA sequence that we never intended to amplify. If this was standard PCR, you would now have a mixture of products. But in nested PCR, we do another round of the reaction, and this time in annealing, we use a different set of primers called internal primers that are designed in a way that they are complementary directly to the DNA sequence we wanted to amplify. And what's the point? Well, if there is in fact a mix of products, these primers will only anneal to the complementary sequences we designed them for, not to the other products of the first PCR reaction. It would be extremely unlikely that there would once again be another very similar DNA sequence that they would bind to, and so as a result, in extension, we get the DNA sequence we wanted in the first place. Our next PCR type, touchdown PCR, is dealing with the same problem as nested PCR, but in a different way. Touchdown PCR modifies the temperature of the second reaction, the annealing, with the goal of making the primers anneal specifically to the sequence they were designed for. To achieve this, in the first few cycles, the reaction temperature is higher than usual. This is because under high temperatures, it is more difficult for primers to anneal to sequences that are in any way non-complementary. The problem is that as high temperatures make it more difficult for them to anneal, it also makes the whole reaction less efficient. That's why in the next cycles, the temperature is slowly decreased. The logic here is that we should already have enough product from the first cycles, and now we just want to achieve a significant yield of the target sequence. Hot start PCR is once again trying to eliminate unwanted PCR products. During the PCR setup, when we add the necessary components for the reaction, primers can bind non-specifically to each other or to different DNA sequences. DNA polymerase can then extend these sequences, and we can get an unwanted, non-specific product. That's why we use hot start PCR, which uses specifically modified DNA polymerases that are blocked at room temperature during the reaction setup. When we first heat the mixture in the thermocycler during the initial denaturation step, DNA polymerase is unblocked and can then later extend DNA sequences to which primers bound specifically under higher temperatures in the annealing step of the reaction. Finally, we have colony PCR, which is a common technique used during transformation, when we insert a vector, a plasmid with our chosen DNA sequence, into bacteria. The problem with transformation is how do we know which colonies accepted our vector and which did not? Colony PCR deals with this problem in the following way. We pick several colonies and scrape a portion of them with a sterile pipette, which we then add to the PCR reaction mix instead of adding extracted DNA, because the initial heating step causes the release of the plasmid DNA. After PCR, we use gel electrophoresis to separate the amplified products by size. We can then see which colonies contain our vector and which do not. So this is it for the different types of PCR. Of course, there are also other PCR modifications, but I think we covered the most important ones. Thank you for watching, and don't forget to subscribe for more videos.